Hi, my dear students. Myself, Anuj Shishidharan, Academic Director and Biology Faculty of Rankings Entrance Training Academy. Today, I am going to talk about one of the most important concepts come under the topic sexual reproduction in flowering plants. Okay, the topic now we are going to discuss, that is post-fertilization events in plants. As you know, what is the meaning of post-fertilization events? You know, all the events occur after fertilization, what we can say post-fertilization events. You are very familiar. The post-fertilization events are the formation of endosperm, Next one, the formation of embryo, okay, the formation of fruit and the seed. So all these events together called post-fertilization events. So first of all, we are going to start endosperm development. Okay, the process of the formation of endosperm. This is the diagrammatic representation showing the formation of endosperm. You are very familiar. The endosperm actually formed from primary endosperm nucleus. This is the primary endosperm nucleus, which is a triploid cell formed as a result of double fertilization. This triploid cell undergoes mitotic division to form a multicellular structure called endosperm. Okay, this is also triploid. Okay, on the basis of the development of endosperm, it's actually three different types. Okay, the first one is called a cellular type of endosperm. Second one is called a free nuclear type of endosperm, and third one is called a halobial type of endosperm. So first of all, I'm talking about the cellular type of endosperm development. Okay, so this is the primary endosperm nucleus. Okay, it undergoes mitotic division to form a two-celled structure. Okay, so in this type of cellular endosperm development, each cell division of primary endosperm nucleus is followed by cytokinesis. See, first of all, the nucleus undergoes divide and again the cytoplasm divides. So clear. So in all these cases, the cytokinesis occur. That means the cytokinesis or the division of cytoplasm occur from the beginning onwards. Okay, that means the endosperm becomes cellular from the beginning onwards. But in case of free nuclear type of endosperm, look, this is the development of free nuclear type of endosperm. The most important point, this is the most common type of endosperm development in angiosperm, especially in dicot. Okay, in case of nuclear endosperm development, you can see that the nucleus of the triploid cell, it undergoes continuous division. Look, first of all, the single nuclei divided into two, again divided into 4, 8, like that. So there will be no wall formation at this time. Okay, first of all, the nuclear division only occur. As a result of free nuclear division, what happened? So many number of free nuclei develop. Afterwards, or finally, the cellular formation. That means the cell wall formation will be occur. Finally, it should be converted into a cellular endosperm. This type of endosperm development is most common in angiosperms, especially in dicot plants. Okay, and third one is actually, it is called a halobial type of endosperm development. Do you know what is the meaning of halobial type of endosperm development? This is actually an intermediate type of endosperm development as compared to cellular and free nuclear endosperm development. Here, the first division of primary endosperm nucleus is followed by unequal cytokinesis. So what is the meaning of unequal cytokinesis? As a result of cytokinesis, two unequal cell will form. This is the large cell and this is the small cell. Okay, afterwards, what happened? The free nuclear division takes place in both the cells. Here, all the nuclei divide again and again to form large number of free nuclei. Finally, what happened? The cell wall formation occur. This is called a Hillobial type of endosperm development. So these three type of endosperm developments are commonly seen in plants. Is it clear? This is actually a diagrammatic representation of a coconut fruit. A coconut fruit is concerned, it's actually having two types of endosperm, This is called a liquid endosperm. You are very familiar. Inside the coconut fruit, some liquid portion is there. This is actually the liquid endosperm, and the white kernel seen around the liquid endosperm, that is nothing but cellular endosperm. So in case of coconut fruit, it actually having two types of endosperm. Is it clear? This is the liquid endosperm. That means free nuclear endosperm. The cell wall formation not occur over there. But in case of kernel, you are very familiar. This is the cellular endosperm. That means, first of all, it was actually the free nuclear endosperm. Again, what happened? The cell wall formation occur over here. As a result, two types of endosperm will be there in case of a coconut fruit. Is it clear? So now the endosperm development is over. Then one more development you are very familiar, that is the embryo development. Okay, the entire process of development of embryo from a diploid zygote. You are very familiar. Zygote is the product of double fertilization. From this single zygote, what happened? A multicellular embryo will be formed. The entire process we can collectively call the embryogenesis. So clear. The process of embryogenesis having a similarity in both monocot and dicot plants. Clear. The embryo develops micropylar end sac, means micropylar region of the embryo sac. You are very familiar that micropylar region, what will be there? The zygote is situated. Okay, as you know, first of all, the endosperm development occur, and afterwards the zygote develop into embryo. Why? Because the endosperm, we discussed earlier, that endosperm will provide nourishment to the developing embryo. Now, I'm going to discuss in detail how the embryo will form from a single diploid cell, what you call the zygote. You're very familiar. The zygote is actually a diploid cell. This is the product of double fertilization. You remember, especially syngamy. Okay, first of all, a transverse division occur. See, as a result of a transverse division, what happened? It's actually converted into a two-celled structure. Towards the micropylar region, there is a small terminal cell and a large basal cell. This is the basal cell and this is the terminal cell. Okay, the basal cell, otherwise called a suspensor cell. Is it clear? Then again, what happened? The suspensor cell or the basal cell, this is the large basal cell. This large basal cell enlarges and undergoes transverse division. As a result of continuous transverse division, what happened? That should be converted into a 6 to 10 celled structure. Look, this is the 6 to 10 celled structure. This 6 to 10 celled structure, what we can say, this is called a suspensor. See, this is the suspensor. The basal cell undergoes continuous transverse division to form a 6 to 10 celled structure called a suspensor. So clear. When you consider the suspensor, the terminal cell, look, this is the terminal cell. This terminal cell is swollen. This is called a hostorium. Look, this is called a hostorium. Okay, and one of the lowermost cell, this is the lowermost cell. Okay, this is the lowermost cell. This lowermost cell is called hypophysis. See, this hypophysis. Okay, this hypophysis later develop into radicle of the embryo. You are very familiar. On germination of the seed, that radicle should be converted into root system. So clear. So we have discussed the developments occur in the basal cell. See, the basal cell, it undergoes continuous mitotic division, means transverse division, to form a 6 to 10 celled structure called a suspensor. This is the suspensor. Okay, this is the small apical cell or terminal cell. It also divided by means of mitotic division to give rise to a pro-embryo. Look, this is the pro-embryo. This is the globular embryo. Look, globular structure or globular embryo. Then again, this is called a heart-shaped embryo, and this is the final stage or torpedo stage. Pretty clear. This is actually the entire process of embryo development from a diploid cell, means zygote. All the process together called embryogenesis. Is it clear? Now we have to learn the structure of a mature embryo. Okay, children, look, this is actually the structure of the diagrammatic representation of a dicot embryo. Okay, this dicot embryo, it actually consisting of an embryonal axis and two cotyledons. You are very familiar. We can call it as dicot, dicot embryo. It's actually having two cotyledons. Okay, so a dicot embryo consists of an embryonal axis and two cotyledons. Okay, the portion above the embryonal axis or the portion of the embryonal axis above the level of cotyledons. This is the above the level of cotyledon. This is actually called epicotyle. This region is called epicotyle, which terminate with the plumule. This is the plumule. You are very familiar. On germination of the seed, the plumule will be converted into shoot system of the new plant. Okay, look, this is the cylindrical portion below the level of cotyledons. Okay, the cylindrical portion is called hypocotyle. Okay, that hypocotyle terminates at its lower ends in the form of a structure called a radicle. That means the hypocotyle terminate with the radicle. Okay, this radicle later converted into root tip. You are very familiar. On germination of the seed, the radicle should be converted into root system. Did you clear? The root tip is actually covered with a root cap. This is the structure of a dicot embryo. Is it clear? Next one, this is the diagrammatic representation showing the structure of a monocot embryo. Okay, especially in grass family. In case of monocot embryo, it's actually having a single cotyledon. It's actually having only one cotyledon. A single cotyledon is called a scutellum. Okay, the scutellum actually situated towards one side of the embryonal axis. Look, that means the lateral side. Okay, at its lower ends of the embryonal axis, it has the radicle. Look, this is the radicle. Okay, radicle and a root cap. This radicle and root cap enclosed in an undifferentiated sheath called a coleorhiza. Okay, look, the portion of the embryonal axis above the level of attachment of scutellum. This region is called epicotyle. Look, epicotyle. This epicotyle has a shoot apex. Look, this is the shoot apex and very few leaf primordia. Okay, all these things, I mean the shoot apex actually enclosed in a hollow foliar structure, means leafy structure. This is called a coleoptile. Okay, This is actually the structure of a monocot embryo. Is it clear? So in this video lesson, we have discussed the post-fertilization events like endosperm development, then embryo development. Okay, and again we have learned the structure of a dicot embryo and a monocot embryo. Is it clear? So all of you have to watch this video again and again for getting full score. Okay, thank you.